Hello and welcome to Better Biopharma, the official podcast of Bioprocess Online. I'm your host, Tyler Menichiello, and today I'm excited to be joined by Dr. Stevin Zorn, President and CEO at MindImmune, a company whose mission is to stop neuroinflammatory disease progression in conditions like Alzheimer's. Dr. Zorn, thank you so much for being here.
Oh, I'm delighted. Thank you very much for inviting me.
No problem. I'm really excited to talk about Mighty 101, MindImmune's lead asset. So, to open us up, why don't you explain to our audience exactly what Mighty 101 is? Uh, and yeah, I'll leave it there for you.
Okay, sure. In order to say what Mighty 101 is, let me just give a tiny bit of background here. Human population genetics have implicated the immune system underlying or being associated with nearly every brain illness that we know today. Yet the industry hasn't yet caught up with therapies that target the immune system. One particular disease, Alzheimer's disease, has a very, very significant root in the immune system dysfunction. And we believe that Alzheimer's disease is a chronic inflammatory disease that's driven by those cells that you mentioned. It's an innate immune cell called CD11C or CD11C positive cells. And what we've discovered is that these cells, which derive from the blood, traffic from the blood through the blood-brain barrier. They settle around sites of pathology in Alzheimer's disease, that being like A beta or tau. And then they produce a synaptic attack on the business end of neurons where, there's communication between neurons and cells, causing synaptic damage. When you damage those synapses, communication gets slowed down or, very, very largely inhibited. That can prevent symptoms and produce progression of the disease. Now, Mighty 101 was discovered to neutralize a target on the surface of these cells to prevent them from crossing the blood-brain barrier and producing that synaptic attack. And so we're developing this compound as a monoclonal antibody therapeutic, essentially to block the recruitment of these CD11C cells into the brain so it can prevent their pathology or their pathologic effects in the disease, but also in other neurodegenerative diseases. Shall I dig a little bit deeper on the mechanism?
Yeah, please. I'm curious to hear your thoughts too on this kind of shift in understanding clinically of diseases like Alzheimer's as more neuroinflammatory diseases compared to how conventional wisdom of plaques and tangles, and just how you see the mechanism of Mighty 101 validating this kind of shift in clinical understanding.
Okay, that's great. So our core hypothesis derives from some really neat science, and that is that it's been discovered that you can have a head full of amyloid plaques in your brain, which is usually the hallmark of when you have Alzheimer's disease. But some people have heads full of plaques, but don't have any dementia symptoms at all. We call these folks, or the literature has referred to them as cognitively resilient. And there's two key Alzheimer's features that differentiates those that get dementia and those that don't, all of which having the pathology. And those two differentiating features are one, plaque-associated immune cells, these so-called CD11C cells that surround plaques, and two, that these cells produce an inflammatory reaction that can be labeled with a dye, in the case of some of these clinical studies, SMI 312, which shows swollen and damaged neurites in the presence of people that have these cells with Alzheimer's disease, but are absent from those people that have this cognitive resilience. And so it was postulated from this study, which came out a few years ago, and a study about a decade before that, that this human data implicates that there's an aberrant immune cell that gets into the brain and produces this underlying synaptic damage. And what we've found is, and so So let me just back up a second there. So this whole notion of the disease being caused by, propagated, and has everything to do with A beta and tau needs to be rethought because there's been drugs that have been made over the last dozens of years that completely prevent the formation of or eliminate the deposition of outside of A beta, and now we're seeing tau. Yet by getting rid of those proteins, people still have the disease. And so clearly there's more to the disease than A beta and tau deposition. And that has given more credence to the notion of neuroinflammation being like a smoldering fire in the brain that's essentially playing a very significant role. And that's why, in underlying this disease pathology, and that's why we're starting to see this shift away from the main culprits that people have thought, these proteins, and moving into different areas. And MindImmune has discovered, three sets of data that really drives this home. The first is that if we label immune cells in the blood with a fluorescent dye, we can then watch, and then that dye immediately gets destroyed by enzymes in the blood. We can then watch where those cells migrate to. And what we found was that in brains of animals that have these Alzheimer's pathologies, these cells, fluorescently labeled and CD11C positive, will, progressively go into and traffic into areas of the brain of these animals, but do not traffic into animals that don't have these pathologies. So they're being called in as a result of the pathology. And because the dye doesn't cross the blood-brain barrier, the only way that these dye-containing CD11C cells could get into the brain is that they are actually carried in across the blood-brain barrier. That was the first finding. The second piece of that finding was that there's a number of studies that have identified the origin of these cells as having come from the bone marrow. The bone marrow, that means in the periphery, not from the brain. And the dogma, around, neuroinflammation is that if you want to treat neuroinflammation or inflammation that's occurring in the brain, you go after the brain's resident immune cells, which are microglia. But microglia are not derived from the bone marrow. They're derived from the yolk sac. These CD11C cells are derived from the bone marrow. And so the only way that they could have gotten into the brain is from the periphery. And more recently, and that's been done by fate mapping studies where you can look at fate mapped markers from development that carry through life. And if a cell carries this marker, you know what its origin is. And the same fate map marker has now recently been found in human Alzheimer's cerebrospinal fluid in multiple studies that were just published in the last year. So we're seeing the same thing in humans that we're seeing in animals. Second piece of data that we discovered was that we did a genome-wide expression studies of immune pathways to try to figure out which pathways were associated with Alzheimer's disease of varying pathologies. And to our surprise, the CD11C cell pathways came up in the top 8 over and over for recruitment and for maturation. And when we were able to neutralize a target on the surface of those cells in the periphery, we could prevent, and this was like a eureka moment for us, Tyler. If you look at a brain section, it's got these green fluorescent clusters of cells all depositing around cortical areas where you get the pathology of the disease. And when we neutralized this target, we were able to almost completely prevent these green fluorescent cells from getting into the brain. And in addition to that, not only were we able to prevent the cells from migrating or getting into the brain, we were able to stop the synaptic damage that was occurring in these animals within 4 days of antibody treatment. And that was astounding to us. Again, it was, you get a picture of an amyloid plaque surrounded by these red, very, very sick, swollen, dystrophic neurites. And after 4 days of antibody treatment, it was completely gone, indicating that these very, very sick neurons had the potential, and the time to repair themselves and to become normal again. And so that was the human evidence that convinced us that this neuroinflammatory mechanism played a very substantial role in the etiology of the disease. And so we started thinking about how do we develop a human therapeutic that we can try this in the clinic with.
Thank you, Dr. Zorn. That was very compelling argument there for the case to go after this CD11C positive presenting cells. I do want to go into a bit of the process development manufacturing aspects behind Mighty 101, but I briefly wanted to talk about the regulatory path that you guys are on. Um,
Okay.
and, specifically, let's start with maybe, in your opinion, what separates Mighty 101 from other existing biopharmaceuticals and competing approaches? Because clearly, and correct me if I'm wrong, but Alzheimer's disease is a hotbed of innovation. Maybe it's newer to take a biopharmaceutical approach to it, but it's a long studied, tried and true indication here. So I'm curious how Mighty 101 separates itself from other large molecule approaches. Or
Yeah, I think that there's 2 ways that it's differentiated from every other approach that's currently being taken today, either drugs that are on the market, and there are now a few, and drugs that are being developed. There are a number of major Alzheimer's disease risk genes that make proteins that essentially increase your risk of getting Alzheimer's disease. If you get one of these, like TREM2, or two of these, like ApoE, your risk goes up dramatically of getting the disease. And other companies are targeting one risk gene at a time. Companies are targeting TREM2, they're targeting ApoE and trying to develop, drugs that interfere with those. What we found was that nearly every one of these Alzheimer's disease risk genes are actually make proteins that are in these CD11C positive immune cells. And astoundingly, every one of these risk genes regulates a very specific function of these innate immune cells. So these innate immune cells, they traffic into tissues and they mount an immune response. They cause inflammation due to various mechanisms. They sample the antigen, they move it out of the tissue, they orchestrate an adaptive immune response with T cells. They activate these immune cells. They cause phagocytosis to chew up, material in cells. Every single one of those functions that's known for these immune cells is regulated by one or more of these Alzheimer's gene risk factors. And these risk factors in total lend some additional support for why this mechanism is very important as a potential therapeutic for Alzheimer's. The second is, and you already mentioned it, the drug doesn't have to get into the brain. And a lot of drugs that get into the brain have off-target effects. Immunologicals, antibodies, no exception. Being very, very potent compounds, when they get in, they can have off-target effects. And for somebody that's worked my entire career making small molecules, it's really refreshing to work on a neuroscience project where now, instead of trying to get the drug into the brain, we're trying basically to target something to stay out of the brain. So that's really what separates MindImmune. So I think when you think about other companies targeting single gene products, MindImmune is trying to capture the collective risk of Alzheimer's disease by basically keeping these multiple AD risk genes out of the brain by keeping the cells that express them out.
yeah, great. Thank you for explaining that, Dr. Zorn. And you do mention your time at Big Pharma, and I wanted to ask you a bit about how that has influenced your approach to these IND enabling studies and CMC development for Mighty 101 specifically. Uh,
Okay.
but I want to shoehorn in there another regulatory question, which is you mentioned earlier the shift in clinical understanding of Alzheimer's disease to be thought about more of as an inflammatory disease. And I'm curious if you see that shift being reflected among regulators, and how is this influencing your approach to IND?
Um, so I think neuroinflammation is arguably one of the hottest fields in neuroscience, disease biology today. So we're not actually in need to go out and convince regulators or anybody else about the importance of neuroinflammation. This is very, very well known. And so the regulators are really very much on top of this. And a lot of companies nowadays are trying to think about not if they should target neuroinflammation, but how. And so clearly regulators are on top of this. They recognize neuroinflammation as a paradigm underlying disease. And so we don't have to make that case for them for that. They understand that.
Got it. Thank you. I did want to shift a little bit to talk about the CMC and process development around Mighty 101.
Okay.
When we spoke in our briefing call, mentioned how starting with humanized antibodies substantially improved your discovery timelines, and I'm curious if you could dig into that a little more, um, and how the
Yes.
decision, how that exactly happened and what that looked like
Well,
anatomi
yes. Yes, that was a really, really important step in our development of an antibody. Time is money in this business. The pharmaceutical discovery value chain is incredibly expensive, and everything that we and others can do to reduce costs is essential to keeping good drug pricing, for future patients. Using a humanized mouse was a short circuit strategy. The old way of doing this was you'd immunize a wild type mouse, develop mouse antibodies, and then you'd purify those antibodies, humanize them, and then do antibody engineering and purification cycles that ultimately gives you what your human or humanized antibody. By using a humanized mouse model, what we ended up doing was injecting the antigen of our target, which is an integrin receptor, directly into these mice. And what spit out the other end were hybridomas that were human. And we were able to purify human antibodies, full length human antibodies, directly out of these mice. And we were lucky. We found hundreds and hundreds of these human specific antibodies to our target, basically saving us about a year of time from having to do the humanization work, for having to do the purification, the affinity maturation work, et cetera, et cetera. And so that was terrific, and we were lucky to find it. We also, just in case, used non humanized mice. We found another 800 antibodies, but those are all mouse antibodies. And we put them in the freezer for a rainy day in case we ever needed to make one. But I don't think we're going to need those.
Yeah. And do you see a shift, with your peers, across the space in starting with the humanized mice, and this approach that you described, or is that, still a little uncommon and/or uncharted territory?
No, no, no. It's everyone is doing that now. That really is the way to go. So when we first started doing this, there were only a couple of companies that had humanized mice, and you wanted to work with the best ones. Nowadays, there's a lot of companies with humanized mice that are out there. They're all good. Not all, but many of them, are very, very good at what they do. And so that really is state of the art now.
Thank you. Thank you for clearing that up. Um, so Dr. Zorn, I was curious, you were mentioning before how Mighty 101 doesn't need to cross the blood-brain barrier, unlike some other therapies in development right now. And I was curious if that has given rise to any unique CMC challenges, um, any, antibody-specific challenges, and how that MOA kind of affects dosing or yield requirements and just general cost of goods. Do you see it reflected that the mechanism doesn't require a dose?
Yeah, so there's a couple of you asked a few questions in there. The first question, does it give rise to any unique CMC challenges? Actually, I was struggling to come up with a long answer to this, but the answer is no. Actually, because we're keeping the drug out of the brain, It's actually it's much simpler because we don't have to worry about off-target toxicity. And so that's not really an issue for us. Um, and in order to make our job easier, once we got a human antibody, we did some engineering to the antibody, to do some IgG switching. Chose the most stable and the most non-immunogenic form of IgG and incorporated it into the human antibody. And then we modified the FC domain so that it didn't have an effector function, so that we get no effector toxicities associated with that. So in many respects, we've come up with an antibody that has very classical scaffolding elements and sequences that have been tested before, all put together with a unique set of sequences that, based on all the data and information that we've been able to develop so far, should be pretty safe. So no, we're not really seeing any unique challenges, specifically when we went ahead and engineered the antibody to try to be as safe as it can be.
Got it. Thank you. And Dr. Zorn, for those of our audience who may not be familiar with your background, you do hail from big pharma. I was curious if you could share a little bit about your time in big pharma and what lessons you're taking from that time in the pursuit of Mighty 101 approval and development, and how that's kind of reflected in your pre-IND work that you're doing right now for Mighty 101.
Yeah, it's when we were in big pharma, we had every possible skill set under the same roof. And so, from clinicians to biologists to chemists, all working together to think about the end product. Um, from the beginning of an idea to the time that we put it into a patient to the time that we put it, on the marketplace. The most important piece that I learned at Big Pharma is keeping the end in mind. You want to write one of these target product profiles and imagine every possible therapeutic use and way of use and formulation that you want. And then you try to design those experiments, not at the end, not when you have a candidate. You don't figure out your CMC and your tox plan once you have a candidate. Think about all of that when you're building your compound. And that was something that was drilled into us in Big Pharma. In the biotech world, companies are small and scrappy, and we don't have all of that in front of us. But it behooves us to make sure that we have all of those connections, that we're speaking to the right people all the time, so that we're developing our plans. Obviously, that any drug that we make has to be reimbursed someday in the future by insurance companies and other payers. And one has to come up with a plan of how the drug is going to be differentiated in that respect. And we need to have the data to show it. So every animal study, every in vitro study, every toxicology study, every clinical study, all has to be aligned along that spectrum in order to be able to, make adjustments along the way so that we ultimately get the label that we want to be able to promote the drug with the properties that we designed so that it can be successful in patients.
Thank you. I was curious how MindImmune is thinking about manufacturability now compared to a later commercial stage and where you're at in that.
Yeah. So right now we're using, various tools to assess developability, of our lead candidate. And, of each of these different tools that we've used, our candidate has hit every single one of those. The short-term goal is basically to scale up the material so that we have sufficient quantity to get through non-human primate, non-GLP dose ranging, and then the GLP study, and then still have sufficient material of the same bulk batch that we can then put into phase 1 in humans. Um, the later strategy is not gonna be just to figure out how to, just get enough material to do that, but how to scale that up on a commercial scale. For us right now, we're generating cell lines, and we're making antibody through, a biological process. is that process that we're using now going to be fit in the future to make sufficient quantities that we need to test in phase 2 and phase 3 studies later? And that's something that we'll have to figure out. Right now we're developing that initial process, and we need to make sure that it's scalable. So that's going to be important in order to be able to show that we can scale up, the product development, and with the companies that we're working with, or will we need to go to bigger and larger capabilities in order to be able to do that? I don't think so. I think again the scaffold that we're working on is not rocket science. It's just specific to the mechanism of action. So I think it's going to be straightforward.
I'm curious how, because, as a small to mid-sized biotech company, you always have to be thinking about scale and longevity. And you're mentioning how you have to keep the end in mind and think about the scalability of your process as it stands. But I'm sure it's difficult to balance thinking about the future with having to execute in the present. And I'm curious if you could offer any insight to your peers in the
What
audience.
what do you mean by executing in the presence versus the future?
So, planning for scalability while also prioritizing what needs to be done to provide product in the present, and not maybe maybe not getting too caught up thinking and future planning, future proofing at the cost of not optimizing today. Maybe that doesn't make sense. I'm not sure.
No, I think it makes sense. I mean, obviously, we wanna do things as cheaply as possible to get to a critical endpoint, and we don't wanna spend a lot of money, doing unnecessary tasks. But that's why we're developing the cell lines now, to make just enough quantity to test us to get into the early phases of the clinic. Later, when we're gonna need much larger bulk of that, we're not gonna be able to rely on these small batch, handling, bioreactors, and that's why and we're not thinking about that right now. But what we're starting to think about, is how do we scale what we're doing to be capable of doing, a much larger scale development of this material for later phase 2, phase 3 studies. And so, um, we have to think about that, Tyler, but we don't have to do it right now. But we need to have an understanding. So there's a number of different pilot runs in the early phase, and they go through manufacturability, tests as well, that predict whether something will scale appropriately. And if we run into flags, and we haven't yet, but if we run into any of those flags, then we will make adjustments as soon as we can to predict that we'll be able to do that.
Okay. Thank you.
Did I answer your question?
Yeah, no, yeah, you did. It's great. Thank you. I did want to spend a little bit of time talking about the regulatory challenges, if any, with Mighty 101. Specifically, if there are any regulatory considerations or hurdles that are top of mind, or are most relevant to Mighty 101. Are there any safety questions or concerns that regulators need answered before moving Mighty into the clinic?
For any immunological drug, the biggest question is going to be, does the drug cause immunosuppression? And there's no way around that. That's reasonable to be thinking of. Um, and so we have done everything that we can so far, looking at the mechanism of action. We don't think that this is going to be an issue, but it's difficult to predict that. In a test tube. And so the GLP toxicology studies that we plan to run in the middle of next year are specifically designed to look for any type of immunosuppression. We've done knockout studies where we've knocked out the target from animals so that they don't have it anymore. Those animals are fertile. They're viable, um, and basically says that they can still respond to some type of a infectious challenge. So by inhibiting or knocking out this target, we haven't prevented the animals from being able to mount an immune response. So all of that data bodes well. that we're not going to see immunosuppression or immunoactivation, but we still have to demonstrate that. And so that's going to be the biggest regulatory question that we'll need to address as we go into those GLP tox studies.
The demonstration, you mean?
Yes.
Yeah.
Well, to demonstrate safety of the mechanism of action. We don't think that the antibody itself is gonna produce that because of the way it was engineered. You know, we put elements of the antibody together that have never been immunogenetic, immunogenic or immunosuppressive before, so we don't expect that. It's really what about the mechanism of action? And so far, everything points to a basically benign mechanism of action, but that needs to be proved.
Yeah, and in that along that vein, are there established biomarkers that you're going to be using for phase 1? And if so, or if there are unique, biomarkers, how are how are you de-risking those for phase
Yes.
1?
The biomarkers, so this is a very, very exciting and very active field right now in Alzheimer's disease. There's the A beta and the tau markers that appear to be able to identify subjects that have Alzheimer's disease. And so being able to stratify patients that have the disease and that have the disease and are likely to have an immune component of that appear to be doing very well. But they're not yet approved by regulatory agencies. So one thing that's going to be important is that we're going to want to seek agreement with with regulators that these biomarkers that are starting to emerge that we can segment patients with are going to be acceptable. One, as a way of segmenting patients, and two, as a way of looking at disease progression. I think that for Mighty 101, I think what's going to be important is and specific for us is that we're going to want to de-risk our clinical development program very early by demonstrating target engagement so that we can show in the blood, that we can block the engagement of these cells with their target tissue like the blood-brain barrier. And there's very simple mechanisms and test systems that we've developed to do that. Second, that we're going to select a set of very specific efficacy biomarkers that are associated with recruitment of the cells, of synaptic health. And I told you why synaptic health is so important, and disease progression. And to that end, in the last year, actually in the last 6 months, there's been really, really amazing work that's come out in the CSF proteomics world. Some of this data was reported just a month ago in the Toronto Alzheimer's disease conferences, where patients' CSF, Alzheimer's patients' CSF, compared to normal healthy controls, was analyzed in tens of thousands of subjects, and what we're seeing on these so-called volcano plots are elevated recruitment markers, elevated synaptic distress markers, and other markers that we have found on these CD11C positive cells that are being elevated in these patients in their CSF. And that elevation increases with disease severity and with the age of the patient. So it's a very exciting time. And those are going to be the specific biomarkers that we're going to look at. If we see evidence of a reduction in these elevated recruitment markers in phase 1, then in phase 1b, then we'll take that as evidence that we're stopping the recruitment in humans. And if we see evidence of a reduction in these elevated synaptic distress markers, we'll take that as evidence that we're seeing an early sign of efficacy. And one thing that we're gonna need to work with the regulators on is the acceptability of using biomarkers as a proxy for efficacy. Can drugs in the future be approved on the basis of efficacy biomarkers? That's still an unanswered question, but for other diseases, that has been the case. So I have every confidence that as we continue to work on this biomarker front in the Alzheimer's area, and this is really, Tyler, this is a really hot field right now, um, that I fully believe that this is going to be ready for us when we're in the clinic a year from now.
Uh, I really hope, I mean, it stands to reason that if there is positive clinical data, I mean, this could open up a whole new therapeutic approach for Alzheimer's and other
Yeah,
diseases like it. You
yeah.
mentioned about, biomarkers and as proxies of efficacy in other diseases. Are there any specifically that come to mind that are most, I guess, closest to, yours being Alzheimer's?
Yeah, yeah. So I already mentioned A beta and tau. Although those are not specifically related to our target, they do identify people that have Alzheimer's disease, and depending on what the mix of those are, whether they are likely to have inflammation or not. Um, Elevated recruitment markers, including like CD18. CD18 is the beta subunit of our specific target, and that's one of the most highly upregulated proteins in the Alzheimer's brain and CSF. Incidentally, the alpha subunit, which is CD11C, is also one of the most highly upregulated proteins in the Alzheimer's brain. And so both of those, we would expect to see changes as potential biomarkers, and there's others as well. On the synaptic distress markers, remember I told you about those resilient patients that didn't have that synaptic damage or synaptic dystrophy. In the Alzheimer's patients that did have that synaptic damage, you can measure that by using that stain, SMI312, which stains those swollen neurite endings and is very diagnostic of that synaptic damage. That same SMI312 stains elements of neurofilament. Neurofilament and its various forms are also showing up in the CSF of and the CSF of Alzheimer's patients in a very, very highly significant way in these volcano plots from the proteomic studies. So we expect some combination of these recruitment markers that I mentioned, NFL, and a variety of other synaptic distress markers that are currently under investigation to be looked at. And we'll be able to look at those in phase 1 because the amount that they go up and the statistical significance that they go up in these proteomic studies is such that we ought to be able to use a relatively small cohort of subjects using an in-subject comparative design to be able to see changes in those markers as early as phase 1. And then once we get into phase 2, then we can look at a whole panoply of additional markers, additional recruitment markers, et cetera, to really, hit the nail on the efficacy measures while we're starting to look at cognition in the phase 2A.
And are there other, because you were mentioning before how it's a hot topic of using these biomarkers as proxies. Are there other notable examples aside from Alzheimer's, indication-wise or, where you've seen some promising acceptance of using biomarkers as proxies for
Oh,
efficacy
sure.
from a
I mean,
from
sure. I mean, just think about metabolic syndrome, for example. A proxy of metabolic syndrome is cholesterol. A proxy of heart disease is cholesterol. So if you have high cholesterol, you take cholesterol lowering drugs. And the reason to take those drugs is that they lower your risk for heart disease, obesity related disease, et cetera, et cetera. So it's incontrovertible now that if you reduce those risk factors by taking medicines, you reduce those diseases. We think the same thing is going to happen with chronic neuroinflammatory CNS diseases as well, that we're going to find biomarkers that are going to be equally predictive. Now, then the thing about the biomarker, for for atherosclerosis, for example, is that you don't feel your cholesterol when it's high. You don't notice, and when it's low, you don't notice. But we know from data now, from years and years of data, that when you lower those markers, you're lowering your risk substantially. And if you get a heart attack, and you haven't already been on those drugs, you're gonna be put on them. Same will be happening in this field and other neurodegenerative fields once we have a strong understanding of those biomarkers and relationship to efficacy.
Thank you, Dr. Zorn. I know we are running tight on time here, so I wanted to give you the chance to answer our hallmark question here on the show, which is and whether this is in your specific realm of therapeutic development and manufacturing or in the treatment of Alzheimer's disease general or the industry as a whole. Dr. Zorn, how do you think we can better biopharm?
Uh, Faster, cheaper, better. Working in, in big pharma, at the time that I was there, the cost of making drugs was astronomically increasing in an exponential way, and the industry and patients were screaming, "We can't keep this up." And so the question, can we stratify our patients? Can we identify those patients that are most likely to be able to respond to our drug therapy is something that we will strive to drive, and is important for the whole ecosystem to get their hands around. Will the regulators agree on a set of stratification markers so that we know who our patients should be? I mean, clearly there usually is not a disease that has one genotypic, fragment associated with it. Usually we have these multi underlying cause diseases. And so we need to be able to think about, how to target just that specific patient, not the one that's got biomarker X, Y, and Z, but the one that has biomarker A and B. And then secondly, by stratifying the patients and then identifying specific biomarkers and getting a regulatory read on the utility of those biomarkers for regulatory approval, we cut years and years off of the development path for drugs. Cutting years and years off of an already very expensive process reduces cost. And that's what everybody wants. I think maybe the third pillar is understanding the disease. And make sure that we have sufficient funding out there to be able to do all the research that we need to understand the brain and all of its complexities and caveats. So when you put all of that together, I think that's what biopharma needs to do to be better. Find ways to figure out who your patients are, target those patients specifically, find ways to know that those patients are responding in a way as soon as possible before you get into long, very expensive clinical trials, and make sure that your hypothesis has a very strong basis in fact and science, so that by the time you get out to phase 3 and you run your first big phase 3 trial and it doesn't work, you haven't wasted all that money, time, and hope. I've lost family members to Alzheimer's disease, and it's personal to me. I don't want to see people suffer from this horrible disease. I don't want to suffer from it. So I've dedicated my career at this point to working towards a treatment, a therapy, a cure. Um, and this is very, very important that we understand the science to do that. And both, I think, the regulators, the biotechs, big pharma, the clinicians, the whole ecosystem, the payers need to come together with those product concepts that we put together and figure out how to do this as an ecosystem.
That's a terrific note to end on. I couldn't agree more. Dr. Zorn, thank you so much for joining me today here on Better Biopharma.
Okay. Thank you, Tyler.
Thank you. And to the audience out there, thank you so much for tuning into Better Biopharma, the official podcast of Bioprocess Online. We'll see you next time.